Hello and welcome, everybody. This is our second annual Millets webinar that is hosted by North American Millets Alliance. And I'd like to ask any of you who are joining us this morning who are not presenters, if you could please kindly mute. That'd be fantastic. We are recording this session so we can share it with our communities later. I'd like to welcome this morning Dr. Vetriventhan Mani and Dr. Dipak Santra, who are globally renowned experts on the genetics and breeding of Proso millet. So if you joined us for our last webinar, we were joined by Craig Anderson and myself, and we were able to really talk about the trends and the market readiness for Proso millet in North America. This month, we are going to take a deeper dive into what's happening at the molecular and genetic level and where we're at globally with Proso millet genetics and breeding. So welcome, Dr. Dipak and Dr. Mani. I'd love for you to just take a moment to introduce yourselves briefly. And for those of you who are joining us, last time we had a panel discussion, but this morning we are actually going to have two separate presentations with a PowerPoint. So that way you can have visual cues to follow along. I would like to start this morning with Dr. Mani's presentation. And if you could please introduce yourself so that people are aware of your fantastic work, that'd be a great place to start.
Yeah, thank you very much, Johnny. So yeah, myself Vetriventhan. You can call me Vetri or Mani, which is easy for you, whichever is easy for you, that's fine. So I am from ICRISAT actually. I am working on millets. So I'm basically a genetic resources scientist working in the Genebank at ICRISAT on millets. Not only Proso millet, but I'm working on almost 8 millets: sorghum, pearl millet, finger millet, the Proso millet, foxtail millet, barnyard, kodo, and little millet. All these millets I'm working, particularly genetic resources conservation and then gem blossom research to enhance the use of gem blossom in crop improvement. My topic of the presentation also the same. I will be basically presenting the gem blossom research and also how we are going to use this gem blossom material in the crop breeding program. So that way I will be focusing largely. Thank you.
Wonderful. And Dr. Santra, if you could please introduce yourself. And given that it's really late in India, we will have Dr. Vetri give his presentation first, but I'd love for you to introduce yourself, Dr. Santra, to our group who's joining us today.
Good morning, good afternoon, or good evening, whoever is there all over the world. I'm Dr. Dipak Santra. I am with University of Nebraska-Lincoln, but located in western part of the state, Scotts Bluff, bordering Wyoming and Colorado. And primarily I work on Proso millet breeding, but my official title is not a Proso millet breeder like soybean or wheat or rice breeder. I'm an alternative crops breeder. So I work on a bunch of crops which can be defined as alternative crops for this area, which is really, really a difficult area to remain in the productive farming. So, and I have been here since November 2008. And if you think about full-fledged breeding, and that's what I'm working on, Proso millet and other things, I am doing part of the breeding, maybe pre-breeding, that kind of things. And thank you so much for nice introduction, Joni, and all the attendees from all over the world on interest on Proso millet. And I would love to interact with you in the future for the sake of Proso millet and specifically Proso millet. Thank you.
Wonderful. And to our audience, if you have any questions throughout these presentations, please feel free to put them in the chat. I will be keeping an eye on them. And that way, while our presenters are presenting, if there's an opportunity to ask that question midstream, I can make sure we get that answered for you. Thank you. So go ahead, Dr. Mani, we'll have you present first.
Yeah, thank you. So allow me to share my screen. I hope this is a full screen, right? Yeah, okay. So first of all, thank you very much for giving me this opportunity to present some of our work, particularly what we are doing at ICRISAT in Proso millet. But ICRISAT, we are working on different crops like millets and also legumes also. Today, I will be focusing on Proso millet genetic resources and conservation research aspects. So you know very well, the year 2023, we celebrated as the International Year of Millets. The millets means it's not a single crop, it is multiple crops belong to the family Poaceae. And this includes the major millets like sorghum, pearl millet, and also sometimes finger millet also called as a major millet. In addition to that, there are many other millets are there like foxtail millet, little millet, kodo millet, proso, barnyard, brown top millet, and there are many more millets like teff, fonio, job steer, all those millets are there, bunch of millets. And this brings crop diversification actually. When we try to introduce the millets into the cropping system, we are trying to bring very good number of crops that helps in crop diversification. And these millets are having basically very good adaptation. So basically it is having diverse adaptation in terms of flying to high altitude, tropical, subtropical, and temperate region. And these millets need less water for their cultivation. That's very important aspect. And also they need minimum input for their cultivation and can survive very well in dry condition as well. And if you provide very good agronomy and then input, then it can give better yield also. On other side of the millets, these millets are having nutritional superiority over the major cereals. You just take example of pearl millet, which is having high iron and zinc. In addition to pearl millet, all the small millets and millets having very good amount of iron and zinc. And then going to finger millet, which is having high calcium content, which is almost 3 to 5 fold higher calcium content than the milk. And you can use this in multiple ways. Millets, you can use it in a multiple ways. And these millets are having high fiber content, protein content, and also high antioxidants. And then coming to Proso millet, you know very well about Proso millet. Proso millet scientifically known as Panicum millesium, and then it is a allotetraploid crop with a chromosome number of 36, and then genome size is 923. And we will be talking about genome sequencing and then genotyping aspects also in this presentation. And then actually this millet, Proso millet, was originated in China almost more than 10,000 years before present, but it spread to other part of the world, particularly Europe and Asia. And main growing countries includes Russia, China, USA, India, and Korea, and many other Asian countries also. And I briefly told about the nutritional profile of these millets, like having very good protein and also very good minerals like iron, zinc, magnesium, calcium, and other things, and also balanced vitamin profile also. And importantly, this is gluten-free. And the other side of the agronomic importance of these millets includes these Proso millet having drought tolerance and can be grown in marginal lands. And the interesting thing of the millet is short duration crop. Among all the small millets, all the millets, this Proso millet is shortest growing maturity duration and belong to the C4 crop species, which requires less input for their cultivation. And also they need low input for their cultivation also, okay? And basically if you see the gem blossom, and then you can recognize 5 basic races in Proso millet, cultivated gem blossom. There are 5 races are there. The first one is millesium, and then second one is patentissimum, and then third one is contractum, fourth compactum, and ovatum. And you can see the images here. So the first one here is the image is the millesium, and then second image is in patentissimum, and then the third one is contractum, compactum, and ovatum. So in the same order only I placed the images. And then the differences are basically panicle morphology and shape. So in terms of millesium, these panicles are large and open inflorescence. Like you can see here, the millesium panicles are large and open inflorescence. And then in patentissimum, this panicle also large, but it is slender and diffused panicles. That is the difference between the millesium and patentissimum. And then other 3 races are basically compact in shape, but the contractum is having compact and drooping inflorescence. But in case of the contractum having the compact and drooping inflorescence, the compactum having compact and cylindrical and erect inflorescence. You can see here that this one is compact and drooping, but this one is compact and erect one. And then the last one is ovatum, which is compact inflorescence and ovate in shape. So these are all the basically 5 races of Proso millet. And global status of gem blossom, if you see, there are more than 7.4 million gem blossom accessions are there, which are conserved in more than 1,750 gene banks. Almost 25 to 30% of the gem blossom, mostly unique gem blossom. Among these, 10% of the gem blossom largely the wild crop relatives, but among the total gem blossom, almost 11% of the total gem blossom conserved or conserved in the CGR gene banks. Coming to this another database, which is a very important database called VIEWS. VIEWS means World Information and Yearly Warning System on Plant Genetic Resources for Food and Agriculture. And in this database, those who are interested to understand the global status of plant genetic resources and then understand the country-wise and the institute-wise, how many gem blossoms are stored in different places, all those informations are available in this, though it is not complete because this database contains only sorghum, pearl millet, proso millet, and finger millet. For proso millet, information is there, and finger millet also information is there in this database. But for other millets like little millet, kodo, barnyard millet, and other millets, information is not yet added in this database. But still, it provides some information about the genetic resources status. And this is another genetic resource status PGR website where the researchers and then the students can look into the gem blossom availability in different country as well as different crop wise. Also, you can see. Coming to the global status of millet gem blossom. So these tables provide the global status of different millet crops. And take example Proso millet. The globally, if you see, there are more than 29,000 gem blossom accessions are there, which are conserved in 52 gene banks. And ICRISAT gene bank, we have 849 gem blossom, which is assembled from, or which is originating from 30 countries. And then globally, we have 479,000 gem blossom accessions of different millets, including sorghum. And ICRISAT gene bank is conserving almost 81,000 gem blossom accessions of 8 millets. And this is almost 17% of the total global millet gem blossoms. And the ICRISAT collections are very large as well as diverse gem blossom collection. And here you can see the region-wise gem blossom status. Okay, you can see here almost 53% of the collections are from Europe, and then almost 41% of the gem blossoms are from Asian origin, and then some almost 4%, less than 4% of the gem blossom from Americas. And here I provided the institute-wise number of gem blossom available, particularly for Proso millet. Those who are interested, maybe you can look into this different institute. And I just highlighted the ICRISAT where we are conserving 849 accessions. Okay, so this is detailed information about ICRISAT gem blossom collection. And then among the 849 gem blossom, all these gem blossom originates from 30 countries. And majority of the gem blossoms are from Russian Federation, and then followed by India and then Korea. And I told you about 5 races of Proso millet. And then here is the distribution of the different races, the collections. So the Millesium is the largest portion, followed by Compactum, Contractum, Patentissimum, and then Ovatum. And then here I just mentioned country-wise number of collections that are conserved at ICRISAT gene bank. So coming to the gem blossom evaluation, okay, we are conserving the gem blossom, but what is the use if we don't evaluate the gem blossom and then if we don't utilize the gem blossom in the crop improvement? So we just evaluate the gem blossom for different traits, phenotyping also we do, and then genotyping also we do. That way we are adding value to our collection so that the gem blossom will be utilized largely in the crop improvement program. So we assessed the diversity of the total collection that we conserved at ICRISAT gene bank for Proso millet. And then you can see the variations for the days to flowering, which ranges from 26 to 50 days, and then plant height from 20 to 133 centimeter, and then inflorescence length, you can see 3 centimeter to 40 centimeter. And that this is some of, you can see so visually the range of variation for panicle length and then different panicle shape. And then country-wise, if you see the characteristics of the country-wise gem blossoms, the yearly and short inflorescence, yearly maturing, and then short inflorescence are mostly from Russian origin. Late maturing accessions are largely from India, and then short plants, mostly from Mexico, and then tall and long inflorescence are mostly from Nepal. Apart from that, we also assessed the genetic distance and then clustering also. You can see here the entire gem blossom. We tried to do clustering, and then the cluster largely it is 3 major clusters are there. The first one is represent Asian accession, and then second one represent the European collection, so the pink one, and then third cluster is mixture of Asia and Europe. And then there are few accession which are from America that is represented in blue color. So this gives idea about diversity and population structure of the Proso millet. And since the Proso millet was originating from China and Asian region, therefore you can see the clear clustering of this accession. And then from here, then it went to other part of the world, particularly Europe. Therefore, we got the separate cluster of this European collection also. And then there are intermediate gem blossoms also there. And then we also assessed distance, genetic distance between the races also, and then genetic distance among the accessions also. That will be useful when we use this material in the crop improvement program. For example, out of the 841 gem blossom that we are having at ICRISAT, so average genetic distance was 0.261, and then genetic distance ranges from 0.01 to 0.51. And then we identified top 10 pairs of most diverse accession. So when the breeder wanted to do any hybridization, they can also look into the diverse pairs of accession so that they can get the high heterosis also. So here you can see here, these are all the top 10 pairs of accession which is having high phenotypic distances. And then we just assessed the phenotypic distances between the races also. And then this information is here in the table. So this will be useful to understand how each race is within race and among races, how the genetic diversity is there. And then, so we have 841 gem blossom accession, but to enhance the use of this diversity in crop improvement, we developed a core collection. The core collection is 10% of the total collection. For example, so when we have 841 gem blossom accession, then 10% of the total collection we developed as a core collection. For developing core collection, there is a process is there. So what we did just here, I just mentioned the steps here. So we took 833 accession, which are originating from 30 countries. And then again, we divided the total collection into 5 groups based on the different races. So the first race one is having 539 accession, and then another race having 98 accession, and likewise the different races and the number of accessions. Then we used almost 20 morpho-agronomic characteristics, and then we performed diversity and cluster analysis of each group separately. For example, 539 accession, we did clustering, and this one 98 accession also we did clustering. Likewise, all the 5 groups we did diversity analysis and then clustering also. And then from each cluster, so again, almost 10% of the accessions were selected to make the core collection. So from here, 10% of the accession. From second group of accession, we took 10% of the accession. Likewise, we made core collection, which is almost 106 accessions. This 106 accession represent the total collection of the Proso millet, which is conserved at ICRISAT Genebank. And also, this is small in number. So therefore, any researchers who wanted to analyze the gem blossom for the different traits like biotic stresses, abiotic stresses, nutrient, or also if they wanted to do genomic analysis also, then this will be one of the ideal subset for to start the research work and then to introduce the diversity in the crop improvement. And that way we have developed a core collection. The core collection is 106 accession. And also we have developed a diversity subset also, which includes core collection and also some of the diverse accession also. Therefore, we made 200 accession. Sometime 106 accession is small in number for genomic investigation and then genomoid association mapping. Therefore, we added more diversity to make 200 accession, so that this is also a kind of subset that can be utilized in the genomic investigation. So further, once we develop the core collection, then the next step is gem blossom evaluation, either the phenotypic evaluation or genomic characterization also. So we started evaluating the gem blossom for different traits. And then initially, so the diversity subset, the 200 accession, we evaluated for agronomic traits. So, for example, days to flowering and then plant height and inflorescence length, under seed weight and grain yield, like that. We assessed for the different agronomic performance of the 200 accession, and then also grain nutrients. So the Proso millet is having very good protein content, but there are very good range of variation also there among the cultivated gem blossom and also the released varieties. So to enhance the protein content, then we need to identify the gem blossom which is having high protein also. So that way, this will help, these studies will help. So from this study, we identified 18 accession which are high yielding. So almost 1600 to 2300 kg per hectare, these accessions producing at ICRISAT location. And maybe it may differ with the different place according to the growing condition. And these are all the list of accession. And then we also identified some of 10 accession which are having higher seed weight, under seed weight. So here, these are the list of accession which are having higher seed weight, which is almost 0.6 to 0.66 gram under seed weight. And then there are accession which are high yielding as well as higher seed weight also. So we identified 2 accession which are having it, which produced a higher yield as well as the grain weight is more. And coming to grain nutrients, there are 12 accession which are having Fe iron content of 63 to 73 milligram per kg, and then zinc content also, and then 40 to 46 milligram per kg, and then calcium content also, you can see the range here, and protein content. So the Proso millet, among all the millets, Proso millet is having very good protein content. Almost 19% of protein is there, but it varies from 10 to 19 or 20 percentage. And 27 accessions are having higher protein content ranging from 16 to 19% of protein also we have identified. And there are 28 accessions which are having 2 or more grain nutrients also. That way, it will be, when we use that gem blossom, then we are bringing the 2 or more grain nutrient into the system. So here, there are accessions which are rich in iron, zinc, calcium, and protein also. So these accession, for example, 2069 and then 2076, and this another one 2537. So these accessions having high iron, zinc, calcium, and protein also. So this type of combination of accession with the combination of grain nutrients are so much important for biofortification breeding program. Apart from agronomic traits and then grain nutrient traits, but we also assessing the Proso millet for drought tolerance also. The same subset, we assessed the subset of Proso millet for drought tolerance in summer season at ICRISAT. And then you can see here the blue color one, the grain yield of the assessed gem blossom under well water condition. And then this pink color is the orange color is the grain yield under the drought stress condition. You can see the under well watered condition. So this is the yield, and then under the drought stress condition, so the relatively low yield also we noticed. But there are accession which produced higher adaptation under drought condition than the well-watered condition. So such type of gem blossom also available, so that we can choose the better right gem blossom, right variety for the right ecologies. In addition to that the previous study was in field condition. Then we also assessed the Proso millet gem blossom for drought tolerance under the lysimeter study also, you can see. And here we studied the drought tolerance of the Proso millet. And then also we are studying the environmental adaptation in the LysiScan also. This is lysimeter facility at ICRISAT, and then this is the LysiScan facility at ICRISAT. We are assessing drought stress and heat stress tolerance, and then so that we will be identifying gem blossom which are having a high adaptation to drought and heat stress also. That way we can utilize these materials in the in the breeding program. And since I told the Proso millet is having high protein content, we wanted to assess the protein content of the entire gem blossom that we conserved at ICRISAT for protein content, so that so we can do a lot of genomic investigation and then QTL mapping or other aspects. And so initially we assessed the protein content of almost 100 gem blossom, which includes released cultivars in India. So these are all the orange color materials are the released variety. And then protein content of this released varieties starts from almost 11 to 14%. And the gem blossom here, you can see almost 9 or 10 to 19% of the protein is there. So this is wet lab data, and then we are trying to build a calibration using the NIRS, and then we will be screening the entire gem blossom using the NIRS calibration for the protein content. This is the idea. This is part of one of my PhD student who is also developing population, biparental mapping population for understanding the genetics and then the mapping of the productivity and then protein trait also. Coming to the genomic resources, there are different millet crops are there, and you know very well. And then genome sequences of these crops also published. And then for the Proso millet, the genome sequence of the Proso millet was published in 2019. And before publishing this Proso millet genome sequence, we genotyped a diversity subset that was conserved at ICRISAT Genebank using genotyping by sequencing approach. And then we assessed the population structure and diversity. And later, once the genome sequence is available, then we mapped the SNPs to the genome sequence, and then we performed diversity population structure analysis and then genome-wide association mapping also. This figure shows the cluster diversity of the Proso millet subset, and then this image shows the diversity within and among races. So here you can see the diversity of Millesium. And then this is diversity of the compactum, the patentissimum, ovatum, and then contractum. And this image shows the mean diversity and the range of diversity among the races. This is between the races like Millesium and compactum. Likewise, these are all the among races and then within the races. So how much diversity is there? For example, the ovatum is having less diversity as compared to other races. So this gives overall idea of how diverse these different races and then among races also. And then we used this genotyping information and then agronomic and grain nutrient data that we generated over 2 seasons for genome-wide association mapping. And we found 40 marker trait association for agronomic and grain nutrient traits. For agronomic traits, we found 34 marker trait association. And then for grain nutrients, we found 6 marker trait association. And then since the environment varied, so since the performance of the genotype in 2 seasons varied significantly, therefore we try to identify the marker trait association which are significant in both the season. Therefore, the number of SNPs which will be so much useful here. And also you see here, once we perform the genome-wide association mapping, it is also very important to identify the favorable allele and their phenotypic value, because a lot of studies we see many hundreds of SNPs were identified which are associated with a particular trait, but how to utilize that information in the genome-assisted improvement of the particular crop. So therefore, what we did, then we try to identify favorable alleles of the different significant marker trait association. So here you see, for example, plant height, the phenotype or the gem blossom with this allele A having tall plant than the genotype with the alternate allele. So similarly, in 3 seasons also the same way it behaved. And another one is for the days to flowering. So for days to flowering also we found there are genotype which we found the allele A, the gem blossom with allele A having late maturity than the gem blossom with the alternate allele. Similarly, you can see different years also the same behavior we found. So this is one of the marker trait association for days to flowering. So this is the favorable allele. For example, those who want to identify the late maturing Proso millet, then they can use this allele in the genomic assisted selection. And then here, this is for the inflorescence length. So genotype with these alleles, for example, G and E are having higher inflorescence length than this one. Therefore, one can use this particular allele of these SNPs for use in the genomic assisted improvement. And this is for the protein content. Protein content also, the genotypes with the allele A having higher protein, so that one can use this allele as a favorable allele for selection. So in addition to that genome-wide association mapping, we also developing biparental mapping population. And then we have developed a mapping population for protein content and also for the productivity triads. And this is part of one of the MSc student who is currently working with Dr. Dipak Santra. His name is Shiva. And then we assessed the diversity and then genetics of the triad, and then we understand the inheritance of different triads. And we are developing the population. Currently, it is F4 generation, and then we will be genotyping this population, and then we will be doing QTL mapping for protein and productivity triads. Once we do this type of activity, and really it will be helping to improve the Proso millet using genomic informations. And in summary, so we have diverse collections that are conserved globally, and then those who are interested, they can get germplasm from ICRISAT also. And then we have developed a core collection, and also we have developed diversity subsets also. And then ICRISAT collection, we assessed the germplasm for grain yield, grain nutrients, and also assessed for the drought stress, and then heat stress tolerance also. And then we genotyped the subset for genotyped the subset, and then we performed genome-wide association mapping for important traits like grain nutrient and then productivity trait. And then we are developing the biparental mapping population for genetic investigation of productivity and protein traits, and then QTL mapping. Ultimately, the idea is introducing new variability in the breeding program can enhance the crop yield and broaden the genetic base of crop cultivars. So that is the key idea of this presentation. So those who are interested to get germplasm from ICRISAT, you can use this email and write to this email. And also you can go to ICRISAT website or Genesis PGR website. From Genesis PGR website, you can directly place your order also. So this is for your information. And once again, I would like to take this opportunity to thank the organizer for providing me the opportunity to present some of our work. So thank you very much.
That was wonderful. One of the questions we did have was from Gary, our own NAMA, Gary Weitzcraft. Gary, would you like to ask Dr. Mani about the photo period and height?
Yes, I was curious, the different heights. I wasn't aware of that wider range, but how does photo period affect height? Meaning, can you take a short variety in Mexico and move it north to the northern latitude and have it actually a medium or a tall variety?
Yeah, actually, since the Proso millet is a short maturing crop, the photo period is having less influence on this crop, but the performance, agronomic performance of the crop will change when place to place, ecologist to ecology, the agronomic performance will change. But the photo duration of the crop is really short duration. For example, 60 to 80 days, this Proso millet maximum. So the duration will not affect much, but the agronomic performance will have the great influence.
Okay, thank you.
That was great. Do we have any other questions from the audience?
Gary, I just have one comment about that one. I have seen whatever tall varieties we did find in the field, 2-3 different locations across the years, and when we tested in the greenhouse, we kept the same photo period, but we just did not limit their resources like water or nutrients. So when we kept on watering, they just went to 7 feet. I had few germplasms which went to 7 feet, and their degree of height remained the same. Trend remained same in the field. They are not 7 feet because of limited resources and short growing season in the outside, but they remain 5 feet or 4.5 feet compared to standard variety, which are 36 inches or sometimes 30 inches. So the trend remains same. I think the major limitation probably is that growing duration, and that at least our location is that growth is halted by short growing season and lack of moisture, that kind of things. That's my observation. I just wanted to throw that.
That's absolutely amazing, because that tells me, as you take varieties that you're developing in Scottsbluff and move them to, say, central Illinois, you may have a completely different look at that particular variety, meaning that it has the nutrients and moisture, for example, and may be significantly higher than what you experience.
Yes, and I have tested at Lincoln 2 years, my germplasm set, and again, average height is significantly higher at Lincoln, and that caused quite a bit of lodging. And because the moisture is not limited at all, and that caused lots of problem like lodging, and then fungal disease, bacterial stick diseases, lots of diseases of what I never seen in the West. So that I have observed in the field in Lincoln also. So what you said, that is absolutely correct.
Well, you have two other issues going on. One is solar radiation, meaning you have more solar radiation at Scottsbluff dryland than you would at Lincoln. And second of all, you're at 3,000 foot higher elevation than you are at Lincoln's, roughly.
That's true. That's true. And the question probably I would request Craig, what you guys observed in that side, like Iowa. You know, you have been testing in Iowa. Do you see similar kind of things?
Yeah. So, Santosh is developing the varieties, in the lab, and then we're doing our hedgerow nursery in Ames for the initial evaluation. And what I think that we've noticed is that the maturities tend to stay fairly similar, but you definitely see, a different type of plant growth because of the available nutrient and moisture. And then the yield experience is almost 3x, what the traditional Colorado yields would be. But I would say in general, the varietal appearance and some of the main agronomic conditions actually are quite similar, whether it's in Colorado or in Ames, Iowa.
Yeah, actually the Proso millet respond very well to the fertilizer, nitrogen. Once you apply the nitrogen, then it gives very good performance. So it is, we also observed that.
Great, good discussion. Thank you. So if you have any other questions, but perhaps at the end of the call, we can revisit anything specific to Dr. Vetri's presentation. But I'd like to move on to Dr. Santra's presentation now too. Are you ready, Dipak? You're muted.
I'm unmuted. Can I share now? Yes, I think I can share. Vetri is still there, I guess.
Oh yeah, let's. Okay, there we go.
All right. I'm going to share what I have been doing in the Proso millet breeding and genetics. And genetics means it includes some genomics also. And the guys who are listed in the presentation, they are the people who contributed in today's presentation in different way. Rituraj Kant, who has been with me for several years. He did my PhD with me. Now he's postdoc. James Snobel, my good friend and collaborator. He has been helping in the genomic side. Devin Rose and Dr. Ui Zhang, they have been helping in this little bit of food science and product developing some small scale research. I will share. Dr. E on biosystem engineering on phenomics, and her graduate student, Mr. Zhao. So I just want to recognize those. So today's presentation is just not contribution from my lab. It is in collaboration with this bunch of people. So outline will be, overall goal is developing high yielding, high performing varieties for my target location in the central high.
Is anybody else having trouble with the sharing?
I think you need to share the slide.
Ah, okay.
Yeah, we're not able to.
What did I do? Okay. Okay. I'm sorry.
There we go. It's coming.
Are you there?
Oh, we have the whole screen view now, the whole desktop.
Okay, let me see. Let me stop that and then
Okay.
go back.
I think if you double click it, you might have it.
Right. Yeah. Is it better now?
Yes, perfect.
Okay.
Thank you.
So these are the people who contributed my work on what I'm going to present today. Rituraj, who has been PhD student, now he's postdoc. Professor James Snobel, my good friend and colleague at UNL, and he has been collaborating on genomic side. Dr. Rose and Zhang on some of the food science research, what I'll present here. Dr. E and her student, Mr. Zhao, on the phenomic side. So these are the people who contributed in different way in my today's presentation content. So overall goal for the Proso millet breeding, of course, developing high-yielding, well-adapted varieties for my target location here in Central High Plain, particularly in the Western Nebraska, Eastern Colorado, and Western, and then South Dakota part of that. So briefly, I'll talk few slides about the background, and majority will be Proso millet breeding, genomics, phenomics, and value addition that side, and little bit of that my program's impact on the Proso millet industry in this part of the country. So I am located here, Scotts Bluff, and belong to University of Nebraska-Lincoln, which is in the east, so about 425 miles west. So there is no similarity between Scotts Bluff production condition and Lincoln. So if you think I am with UNL means expecting Lincoln environment, no, it is totally different. It is almost like Wyoming environment, Colorado environment. These are the challenges at our location. Semi-arid high plains means it's basically high altitude desert, very limited moisture. And these kind of things happens pretty often. And the limitation of water is this. Our need is this yellow curve, and available moisture during the growing season is this red curve. So the significant deficient. Because of that, the traditional production system in the dryland system, winter wheat-based wheat fallow system, means 1 year there will be wheat harvest, and that year it will be planted in the fall, and this section of the field will remain fallow. And this will be, say, this was harvested 2024, and this is being planted for 2025, and this will remain fallow 2025. For traditionally, it's 2 crops in every 2 years, like wheat fallow. That's the traditional because of limited moisture. So that's the place I play a role. If I can develop some well-adapted short duration variety which can replace partially this fallow, probably that will improve toward increase the overall farm productivity and farm sustainability. For example, millet could be 1 crop, pea could be another crop. So instead of 2 crops in 4 years, you can get 3 crops in 4 years. Or if you have magic and develop continuous cropping system, then every year you get crops. So your potentially, theoretically, you are increasing your profit potential to double from 2 crops in 4 years to 4 crops in 4 years. So that's the place. And because of that, as I said at the very beginning, alternative crops, I work a bunch of crops. So I just grouped the 4 different quadrants. First quadrant is I called as ancient grains. Proso millet is, and then other crops, amaranth, buckwheat, I tested here and there. And then oilseed crops, camelina, canola, sunflower, and then pulse crops, field peas, garbanzo beans. And sometimes I tested some mung bean, that kind of things. And the last group is industrial crop, other crops like mint, industrial hemp. But today I am focusing on Proso millet and sharing few things. Why Proso millet? Proso millet is the best alternative crops as a rotational crop in this area. Because of this, shallow root system adapted to this area, very, very drought adapted crop because it uses least amount of water among all the cereals. I think this is the only cereal which can produce grain to grain when no other cereal can survive and produce the grain. This is one example. 2012 famous drought in the Great Plains. And this is in near Sydney, Nebraska, 1 mile apart in Sion County. This is Proso millet field, and this is corn. I don't think there was any single productive kernel here, but there was a decent yield, about 25 bushel per acre yield. And that is primarily because of this. This is 110 years old study from Akron, Colorado. This x-axis has different crop and y-axis has the amount of grain produced per ounce of water or gallon of water. Yeah, ounce of grains produced per gallon of water. If you see that, wheat, barley, maize, sorghum, foxtail millet, Proso millet is certainly is the champion. That means Proso millet produced the highest amount of grain using 1 gallon of water. 110 years old study or more than that. This is the same similar study from the same location. This is probably better representation. Proso millet starts producing grain at little more than 4 inches of acre water, whereas corn starts producing kernel at 8.25 acre inches of water. So when corn starts producing kernel, millet reaches approximately 75 bushel. That's the potential. That's the beauty of Proso millet in this kind of environment where water is a problem. So it has been well documented. Whatever mechanism would be, if the water is a problem, Proso millet is your cereal crop. Briefly, this is a little bit old. The number probably not matching with today's number. These are old slides, but it is very localized in this area. Colorado is the number 1, then Nebraska is 2nd, South Dakota, and North Dakota, then little bit in PNW is going there, and Minnesota, but primarily 90+% Proso millet acre comes from this area, high plains, Colorado, Nebraska, and South Dakota. Now, our university, this program is that pioneer in the Proso millet breeding, in public breeding, even private breeding when it started. 1965 it started in the same place here. And agronomist Dr. Grabowski, he started and doing some simple selection. He released that. Then Dr. Nelson came after that. He worked almost 17 years and released 4 different varieties, followed by David Baltinsberger, who is now Texas A&M. He developed few varieties, again, 4 varieties, many of them are commercial production. Then I came on board in 2008 and I started working on different aspects. These are the historical Proso millet variety in that US. I grouped into 2. That bottom half are the primarily selection, single plant selection from the germplasm that time was available in the US Genebank or many research program. And then top half are through breeding, crossing followed by selection. So this today's commercial varieties, like in public breeding setup, other than some private few varieties available from Dryland Genetics, like Dawn, Ardliver, Huntsman, Sunrise, Horizon, Plato, these all came from University of Nebraska's breeding program. And they are still in the commercial production system even today. So primarily, I am working on traditional Proso millet breeding with the intention to develop high-yielding varieties through traditional breeding approach. And the side, I am trying to develop some breeding tools as well, like primarily germplasm and then genomic tools. So, and this is the traditional breeding panel, as probably all of you are familiar with. In a self-pollinated crop, you make a cross, then develop hybrid, and then expand the population, F2, F3, F4, and then start hetero selection at F4 level. And then you filter it out like a reverse funnel. And then after probably 10, 11, 12 years, you release a variety through multi-location testing, what I typically do in these 3 locations. In Nebraska, a couple of locations here, and then 1 location in South Dakota, another location in Akron. So through multi-location variety trial, I developed the variety. That has been the strategy. So first variety 2015, what my predecessor started, I finished that and released that, is the first public waxy millet variety in North America. Waxy means stickiness. All the varieties are non-waxy. They are not sticky because of that composition of that starch. What is the variety? I forgot the starch, two different type of starch. Anyway, this is a sticky millet variety what I released in 2015. And then I continued doing that in addition to other crops and with little funding I had. And this is that probably 4 or 5 different potential new varieties, which I will be releasing at least 1 or 2 this year, followed by a couple of more next 1-2 years. I just want to show you that snapshot of this multiple years of data. These blue circle, those are all Czech varieties. See, their highest yield was 1,484 pounds per bushel. And all these 5 Czech varieties above that. So they are statistically significantly higher yielding over multiple years. And these will be released few in this year, at least 1 or 2, and then followed by few more. And in a public varieties testing site, my colleague, Professor Creech, he conducts that. This is the 2014 variety testing result, other than mine, is average of Akron, Colorado, and Sydney location. If you see the left column, these top 3 are from my program. Then DLG is 1 breeding lines, experimental line. Then DLG is like Dryland Genetics public released variety 240, and then again another one mine. And all the Czech varieties are below all these lines. So it is not only at my own testing, those my breeding lines are performing well, but also in the public testing settings. This is 2014, 2013. I put only 2, and again, these lines, top 1 is the number 1, and that remained. If you go back, is this, sorry. If you see that 93, that ranked here. And then 101. So basically the message is those 5 varieties, they are doing well and they will be released, and they are all traditional starch content like non-waxy. Now I'm going to share a few slides quickly about some genomic tools I have been developing based on the germplasm what we have in US Genebank system, very similar what Vetri presented. US germplasms. This is the data I'm just showing based on that. David Brainerd collaborated there. Based on his input, we selected 85 different germplasm from several countries, and we just did based on morphological study in the field testing. These are their overall, if you see the ranges of different traits, here is the different lines, number of lines from different countries, and which are not adapted, as Vetri also presented, that all the Indian origin germplasm are very, very late, and they did not flower here within our short growing season. Nevertheless, whatever flowered, their flowering time ranged is significant. Same thing, the height, then inflorescence length, and 1,000 seed weight. This is not any different what Vetri presented. The message is there is a significant variability present within the Proso millet germplasm resource, whether it's a ICRISAT or China or Europe or US. And there is an opportunity to select whatever way you want. You want late or early line, large seed or small seed, tall or short. So that's the message I'm trying to give. And that is the gold mine for breeders, because if there is no variability, you cannot select for crossing. These are the different the germplasm names, and like early, late, tolerant, susceptible, and that kind of guides me in selecting the parents from the crossing for the crossing block. This is that different cluster analysis of different groupings based on the phenotypic data. And we notice it is not illustrated here. Similar kind of situation we have seen. Asian lines, they cluster together. And then North American line, at least in my setup, they found quite a bit of cluster. And other groups, they are mixture of lines from all over the world. This is based on the SSR marker, similar kind of things we have noticed here. This is based on morphological marker. This is based on SSR marker, simple sequence repeat marker, which is an old-fashioned DNA marker. And we also did SNP marker, like modern marker, similar kind of situation grouping we saw. These red lines are from all Indian lines, like Indian subcontinent. These are primarily from North American because that's the cultivars. And these two groups are mixture of European lines, some of the Asian lines, and also North American lines. So that is based on SNP. So we have done morphological based cluster analysis, SSR marker based, SNP based. Overall trend remains same. Of course, there will be some exceptions, basically based on the nature of marker we used, but Indian lines or lines from the Indian subcontinent, they cluster very tightly. North American also very tightly. So that is a good thing. That is a good thing. That means our system is working and across the marker, they are genuine. And then that is our diversity analysis. This is, I'm going to talk about mapping. How to identify those genes which are important for breeders for the selection. Now, traditionally, we grow thousands of population in the field. We measure their phenotypic trait traditional way. When they are flowering, measure their height, measure their yield and seed size, and you select. But in the mapping, you identify the gene for those traits and select those genes in early generation before even taking those material to the field so that you increase your breeding efficiencies. So that couple of strategies, biparental mapping population and natural mapping population. So I'm just going to show our result when 2016, when the Dryland Genetics millet breeder, Dr. Rajput, he was the first graduate student, and this is from his work. So this was the first Proso millet map, genetic linkage map we published. And all these triangles, they indicated that different genes. I do not know I have table or not, but different agronomic traits we measure and we could map on the Proso millet different chromosomes. That is way back before that Proso millet genome was sequenced. Just to give an example, one is the grains per panicle. We mapped that on one linkage group, and this is the marker we identified there for lodging, for grain shattering, and for plant height and panicle length. These particular two traits, lodging and grain shattering, is extremely important for our farmers here. So any work towards identifying the major genes or genetic dissection of those two traits will be tremendous contribution to the Proso millet breeding program. And obviously that needs extended collaboration, not only within US, but also across the globe. Genome-wide association mapping. So this mapping based on the biparental mapping population, and its application could be limited because you are creating that linkage block and you are mapping the gene. So there is a limitation. But when you are using natural mapping population, probably applicability is much better. So we did that work also, a subset of population. We tried to do that, 71 germplasm from the US Genebank, and about 11,000 different SNPs. We used that to do the analysis and with the goal to identify marker on natural population, which has been evolved over thousands of years. So their linkage block, they are already adapted, they are fixed. So whatever marker you identify using GWAS approach is more applicable all over any breeding program for the indirect selection. So we did that subset of population, and here is the result of 18 different chromosomes and the SNP distribution. Basically, the red indicates highest number of SNP marker within these regions per MB, whereas the green means least amount. So obviously, based on the nature of the genome, their distribution varied significantly. That no color means there is absolutely no SNP identified, at least based on this study, and there is a variability. So based on this analysis and the phenotypic trait we collected in the field, we identified marker, significant marker. And this example is a Manhattan plot and just showing the three different characters: heading date, lodging, and 100 grain weight. The way to read this, the x-axis has a chromosome 1 to 18, and y-axis has that log value, threshold level. Any marker, the dot you are seeing, if it goes above that horizontal line, they are the significant marker associated with the trait. Like for example, here, chromosome, this is 18, 17, 16, there is nothing. In 15, there is one marker. 18, there is one marker. And chromosome 15, I think this one is one marker. Same way, this marker, that's the way to read that Manhattan plot. So there are quite a bit of markers identified for 100 grains per weight. This is just basic genomic analysis we do. But for the breeders, take-home message is this. Vetri presented in 3 different traits, probably number of markers. We identified total 20 marker trait association for 5 different traits, like 3 for heading date, 4 for internode and lodging, 4 markers, panicle length 3, and grain weight, probably 6 markers. And this is Vetri is part of this work. We have been collaborating on a limited aspect, but we are looking forward to expand that collaboration with ICRISAT. And this marker eventually should be useful and should be used in the selection in the breeding program, but that needs a different setting, different resources, primarily funding. And public breeding, it is extremely difficult and limited, but I know that like Dryland Genetics, they have been using this approach and they have been very successful in their selection. So there is a place to use that, but for my program, I have not been using anything of these markers in my selection, primarily because of the limitation of the resources. Then phenomics. So I talked about traditional breeding, then developing genomic tools, then phenomics. What is the phenomics? If we want to know when the thousands of my genotypes or plots or single rows are flowering, I need to send my crew probably twice a week during prime time, window time, and they have to go, and sometimes we get limitation based on the weather, based on our crew's sickness, we may miss that opportunity. So alternative is, with the technology we have, can we fly a drone over a plots of 1,000 plots, genotype, and capture that image and develop some computer algorithm and ask the computer, tell me which plot has flower on particular date. That's the approach we wanted to test that. So this, we did that using UAV-based drone. We used 64 different test lines. This is just test of hypothesis. We flew this drone, captured the image, developed the algorithm, and tried to test that in-field data. So we took image-derived heading date, and we got field-derived image heading date. And then we compare to test which algorithm or which model works the best. So this is the way it works when in image analysis. You take the image, then you cut it, and then you identify that using that program and tell that how much of this single row has flowered, as it is indicated by small red box. And then we develop a model. So based on this model, we are able to go to confidence level 95% and above to correlate image-based phenotyping and field-based phenotyping. So we felt pretty confident about that, and this can be applied to other traits. For example, plant height, maturity time, lodging, all these things, and that can be done. That can be done. So I just wanted to show it is not only breeding genomics, but we are also doing some phenomics. We have done that. We are looking forward to expand to other traits. And then as I came in 2008 and 9, I quickly realized, well, there are some varieties, but major limitation for millet industry is the product development, the market. Market is limited by this bird feed industry. Even today, majority, probably now it is 85% of the Proso millets market share is based on bird feed industry. But in globally, Proso millet is primarily used for human food. So why can't we do that? So that's the kind of things I reached out to my colleagues in the food science and technology and decided, let's see whether something can be done to expand that portfolio of Proso millet derived product in the market. So those are the few slides I just want to show what I could do in partnering with those my colleagues. We did some simple grain analysis, just main components of the varieties. So we use those commercial varieties, and starch, protein, fiber, fat, and ash. Not significant different with all these traits, as you expect, because their genetic base is very narrow. We did that before doing any product related food science research. You need to know what are the nutrient composition in those. In that exercise, we did that. And then we expanded that using about 28 different lines, including those check lines. Same thing we did it, protein, fat, phytate, which is one anti-nutritional factor, starch, lignin, dietary fiber, and we did see quite a bit of significant difference. Again, Vetri already presented in his ICRISAT collection how widely, and I did a small set of lines. I did find the similar kind of things. And then we collaborated, this is with University of Kentucky. We wanted to do a little bit of functional assay of Proso millet protein. And the different attributes are like surface hydrophobicity, protein solubility, emulsification, and foam capacity. Why these are important? Now, Proso millet variety may have 15–18% protein, but how do they function when we are trying to develop some product? So that's the question Bode, who is my collaborator at University of Kentucky, he addressed. And in any cereals, there are 4 basic fractions of protein: albumin, globulin, prolamine, and glutenin. And albumin, globulin are the main components of any cereals, as you see here in the Proso millet. You see all these 4 fractions, they differ significantly with respect to all these 4 traits, whether that is a surface hydrophobicity or solubility or emulsifying capacity or foaming capacity. Significant difference with all these. These are like varieties we used, Dawn and Plato, and we did different. And here, again, same thing, those 2 varieties in different aspects. The message is, we are not stopping at the, okay, we found our majority varieties are 11% protein, and we do have germplasm with 14% protein or 18% protein. But how do they behave? Will be there a significant difference with respect to protein functionality among those germplasm? That's the one we wanted to test, and we did find that, and we published, of course. And another millet issue with the protein digestibility we noticed in the Proso millet, when the bird eats, it is uncooked millet. But when we try to develop some product using Proso millet, we cook it. What we found when we cook Proso millet, whether you use millet flour or isolated protein, their digestibility significantly goes down after cooking. What it means, although we have 20% protein, but if they are not digestible in human system, probably that is useless, most likely. And that's what we noticed that one, and do not know why. We tried to find out, we identified some motive. That's what we noticed that, because the digestion sites are inside the protein 3-dimensional structure. That's what we found based on that secondary structure of those proteins. How to overcome that? There are different approaches. We definitely can take that. But right now, we are not just going into that direction to develop a Proso millet variety with a better digestibility. Traditionally, it will be difficult. We need much more work testing the digestibility analysis across the germplasm, or modern breeding approach is a CRISPR-Cas technology gene editing system. That can be possible because we identified few targeted amino acid. It can be edited and put in the Proso millet genome. For example, here, this is the panisin gene in the Proso millet. There are in bioinformatics analysis, we identified 4 groups of those panisin genes, and we can target those genes and change the amino acid so that they are not hiding inside the protein, but they came outside. That is one approach we can increase the digestibility of Proso millet protein. Proso millet can be used as efficiently as corn in bioethanol production, whether it's for industrial or even consumable alcohol beverages. So we just tested that time quite about 10 plus years ago. We tested high fructose corn versus millet. What this graph shows, their starch content is going down, both corn and proso millet, they are following same track. As their starch is going down, their alcohol production is going up. So the message here we found, the proso millet is equally useful as a corn, as a feedstock in commercial bioethanol production or in distillery plant where you are using proso millet as a feedstock, as a basic material for whiskey or beer or wine. Gluten-free noodle. We did a small test based on wheat, cooked noodle, uncooked noodles, uncooked and cooked, and here is the proso millet. We did their physicochemical analysis comparison. There is not much different. So proso millet flour can be used to develop gluten-free noodles. I'm sure in the market may be there, but we tried that one and we did find it does work and it is possible. Flakes. This we did not do that. No, we did that. Like we can do like popcorn. We can do popped millet. So there are different avenues we can expand the market. And this is not my expertise, not my job description, but I thought that is important to at least show that industry that the proso millet can be used in different direction, not only the bird feed, in diverse type of food, which is compatible with in the US market system. So that's the type of like overall goal I wanted to collaborate with my food scientists and technologists. And this is the few things I just wanted to share. We did that. My contribution, this is again couple of years old study. Like based on that USDA NAS data, like 2021 proso millet acre was 160,000 in Nebraska, which is approximately 36 million. But compared to 2020 and 2019, so that increased significantly. And quite a bit of contribution probably came from me because I not only research, but I do the millet extension work. And so I have been working with the farmers in this area. Of course, market plays a huge role, but I don't think I should be hesitant to claim some of the contribution in the proso millet industry here in increasing this acre. One specific variety, Plato, when we released 2015, almost we had no commercial production, but 2021, we had about 8,000 acre. And I talked with Leon, now it is 10, I think 12 or 13,000 acre that waxy millet. So those are the few contribution with respect to economic contribution, with respect to academic contribution, what my university bosses would like to see. Primarily, to keep my job, we have published several papers on different aspects, generated some fund over the years from different sources to continue working on the proso millet. This is my last concluding slides. I'm not elaborating, summarizing what my work scientifically, but what next? That's what I'm coming here. I think we have several things we can do to expand proso millet and to make proso millet as a visible climate resilient alternative cereals in the future climate scenario. We need to continue genomics and modern breeding work. For example, we cannot stick with traditional breeding. We need to use these modern breeding tools, whether genomics or phenomics, to reduce the number of years to develop variety from 10 years to 5 years for the sake of farmers, for the sake of proso millet industry. More agronomic research, biomedical research to show proso millet is important for human health. It is not only important for climate health, it is not only important for probably farmers' wallet help or bank account because proso millet uses least amount of water. And when they lose the crops, say wheat or corn, they can get some money out of proso millet, but also human health. Maybe we need to show more visible result from that. Product development. more ready-to-use product development for North American market. Because traditionally, China, India, the way they are using, that's not the way our US people will be using that. So we need to develop more ready-to-use product. And I'm happy to see that is coming, like Joni, and then another industry in Canada. They have started some, and that is I really appreciate your courage to initiate some millet-based industry and help the Proso millet industry. More public-private partnership, like public breeders like myself. We need to partner with a private industry like Dryland Genetics. Food industry, we need to talk like Joni's company. What can we do to address some of the issues what you may have? Public awareness, farmers engagement, and global engagement. Do not have many millet researchers all over the world, whether it's a food science or breeding or agronomy. Very few, limited compared to say wheat, rice, or soybean. So I would say these 4 areas are most important moving forward. More breeding work, product development. In my opinion, product development should be number 1, because if there is a demand, everything else will come. Everything else will come. Public-private partnership and global engagement. Number 1, I have been telling that, and I started working from 2009. We need to expand that market. We need to expand the market, diversify the portfolio in the market, not only bird feed, not only maybe 2% of US Proso millet used in whiskey production. What about different food? I know there is a company, Dr. Scher, I think in Italy, they are specialized in gluten-free product, and they're primarily using Proso millet. So why not in US? So primary limitation probably, who cares Proso millet in US, which is where corn and soybean are the king. In 2023, International Year of Millet, in the UN, when there was a vote, if I remember correctly, US representative voted against Proso millet or millet in general, which is unfortunate, but we few brave people like Don, Joni, Gary. Gary is like a probably living library. Anything you ask Gary, Gary will tell. Gary has the answer. I have no words to express his depth of knowledge and his passion for Proso millet. I think he's a global resource. So we need these few important areas we need to focus, making Proso millet more visible in the global context. And if we really care about millet in general, at least in North America, primary focus has to be Proso millet, because we are already in the market. How can you do better and expand it and get more policy from the Congress in the Farm Bill? Because without their support, probably farmers would not be interested. Idea, for example, if Proso millet has a less environmental footprint, so farmers should have subsidy. If some farmers is using 10% of their farmland instead of corn and soybean, if they are putting Proso millet, there should be some incentive from the Farm Bill, that kind of things. And that is not possible by me directly, but the grassroots level, our stakeholders, whether you are industry partners or farmers, they need to work with the local policy makers, senators, Congress people, and so that what is the contribution of Proso millet, what is the Proso millet potential 10, 20, 30 years from now, where it cannot be resolved in overnight. That kind of things. I'm not asking a big money from federal agency. Can they put even 200,000 year mark for Proso millet research? What they have done in canola research, what they have done in dry pea, lentil, and chickpea research, that kind of things. And that's my appeal or request to all the stakeholders here. Whoever can do, just rub the shoulders of your local policy makers. With that, I'll stop here and I will try to answer if some have some specific questions. Thank you very much.
That was wonderful. I resonate so much with a lot of the passion here at the end of this, Dr. Santra, because it is shocking to me with all the incredible attributes and reasons to grow the market for millet, I've spent 10 years raising money to develop and commercialize products made from millet, and it's been impossible almost. I mean, 10 years, and there's just not an appetite for anything outside of the big five. Unless you're like hyper focused on gluten-free. But in my experience, like there's such a huge role outside of that segment. I mean, like right now we have a pasta product. It's already been piloted. It just needs money to commercialize, and it's a blended regenerative pasta. It's durum wheat, Proso millet, sorghum, and garbanzo bean flour, and it tastes like a regular pasta product. But investors don't have any appetite to help you get that to market, and it's expensive. So it's very frustrating.
Yep. Yep. Like
It's
I
a
have
great
been,
discussion. Yeah.
I have been doing this Proso millet research, almost no funding. Dryland Genetics
None.
is extremely generous. I have been testing their breeding lines in this location. I get some money from them just to pay some basic bills, but that is probably 10-20% or even lower than any breeding program needs. Breeding program cannot be supported just 1 year in, year out. We need 5-10 years. That's why if you see North America, all the major crops, they have either state commodity board or national commodity board who are providing the base
That's right.
sources. And without that is impossible. And I'm getting extremely frustrated, and I'm bootlegging some of the other sources of fund to do my Proso millet work. And that's why you will see that question you may ask, last variety I released 2015, what took so long to release, like reach to a place where I have a better varieties? Funding.
Yeah.
Expenses going up. What is the inflation today?
Yeah.
And funding source is going down. So we are kind of extremely challenging situation. And with the current political environment in Congress, whatever is happening, few program I am trying to submit some proposal. When I go to USDA's website, is blank. They released the RFA, now they are silent. They took out the RFA. So extreme challenge, but challenge is the sign of opportunity. If we work together, if you brainstorm, probably something will come and something has to come.
It's one of what we're trying to do with this webinar series this year, our second webinar series, we had one in 2023, is to help expand the dialogue and deepen the dialogue about how we can develop strategies, interest, programs, and ultimately funding and participation in efforts to expand millets and in agriculture and food in this continent. A lot of potential. I really want to thank you both, Dr. Mani out in India. It's very late where you are, and Dr. Dipak and Santra in Nebraska. You're in the middle of your day along with ours. And I think we'll wrap up pretty quickly. I see, I don't want to usurp Joni here as the moderator, but I do see David Brenner raise his hand.
Oh, yeah. David, are you there?
I was just clapping. It was the clapping icon.
Oh, okay. Good.
Not the raising hand. Maybe somebody else has a more thoughtful comment than that.
I have just one comment to tell connected with that earlier question. What is it? Only Proso millet in the US or any other millet? What is the potential? That kind of things. I was in Vetri's program in 2022, and I went through his nursery, and we had a good discussion. Both of us, we selected a subset of his collection, not only Proso millet, but primarily foxtail millet, finger millet, and barnyard millet. These are the primarily 3 non-Proso millet we selected. And I have been slow bringing those germplasm to US here and testing their adaptability and see what happens. And primarily because of the funding again. Most of my crops do not have major fund. So how many things I can do free, I do not know. But I did not give up the hope. I am continuing. I at least bring those lines here and based on my resources, I can test maybe 10, 20, 30 lines from all those 3 millets. So I'm doing that. Another question is pearl millet hybrid. I know one professor at K-State at Hayes. He has been working on pearl millet hybrid to be adapted in this grain type, human food grade quality hybrids. And last time I talked with him, he said there are some potential new hybrids. They will come to the market, at least he will release. And then commercial production, that is in nobody's control. That depends on the market. So from personal communication, he said there will be some potential short duration human food grade quality pearl millet in the US. And I know somebody in Texas has been working on finger millet, but I believe it's primarily as a hay crop. And foxtail millet for grain, I do not know. But in our area, we have 2 varieties which we have the certified seed, Leon Crisel, but is primarily hay for the hay production. And I have tested those lines. So they do produce seed, but they are not selected for grain like human food grade quality foxtail millet. But they produce viable seed.
Yeah.
So
we dehulled some foxtail millet from Nebraska and tried it, and it was definitely edible. It was good, but it was a little more coarse than some of the culinary ones that I've purchased. But you know, now that hopefully the USDA continues to support this millet dehulling plant in Belgrade, Montana, because they actually are the only ones that I know of that would be open to commercially dehulling these other types of millets. We have somebody planting finger millet in Spokane, Washington this year, a trial to see again if that would be a viable summer fallow alternative in this region where summer fallow is the norm. It's a wheat growing region, and we have huge, horrible air pollution issues with soil blowing away after tilling in the spring. And millets provide, and C4, you know, warm temperate cereals provide an alternative to eliminate summer fallow regionally for us, which is a huge environmental concern. So yeah, we're making headway, but you know, this is like a whole nother great discussion. And I know people probably do need to wrap it up, and I'll quit recording, but I think these are things that we need to put out into the world because this is critical. And like you said, Dipak, the market is in a state of paralysis with innovation because there has been no capital invested in years. It's just been a stalemate. So
we'll revisit some of these topics in the next webinars. We're still putting together the program. We have some ideas, and look forward to feedback on that. We're trying to do this monthly on the third Thursday of every month at the same time, which would be 11 Eastern, 8 Pacific, and around the world.
And Don, just one request. We can do great science. There are great work, great presentation, but can we bring some real policy makers who has some voice in DC in a Congress so that they get to hear what Proso millet or millet in general has to offer to the US agriculture?
I believe there's going to be some effort to bring some policy people. I can't speak for them, but this upcoming conference at Purdue, the GSA Millets Expo, in April, there may be an effort to bring some legislators in as part of the program. So I don't know their program. I can't speak for them, but I'm looking at what they've done in the past. That might be a possibility.
Great.
Excellent.
Anyway.
Well, thank you, everybody. This has been wonderful, and we are recording it, so we will have that available on YouTube in the coming weeks to share with your friends and communities. So thank you so much, everybody. And thank you to our presenters. That was wonderful.
Vetri, it is 11 p.m. your time. Thank you so much.
Yeah.
And I will see you in April, probably.